Great. Well, I think it's almost 10:05, so I think we'll just go ahead and get started. Thank you all for joining us. We're here at the Center of Genes, Environment, and Health. This is kind of our online forum to discuss questions around precision medicine, human genetics, and particularly focused on populations understudied and underrepresented in medical research. And today we're really happy to host Dr. Tim Thornton, who's at University of Washington. Tim got his PhD at University of Chicago and subsequently did a postdoc at UC Berkeley and UCSF with Neil, who's here, which is great. He has been really at the forefront of doing, first, a lot of pedigree-based work, and then integrating the idea of pedigree-based statistical analysis with genetic admixture analysis, and has developed a lot of tools that we commonly use in that domain. And he's here today to tell us about transcriptome-wide association studies in ancestrally diverse populations, challenges, and new opportunities. Thanks, Tim.
Great. Thanks so much for the introduction, Elad. It's good to be back at UCSF virtually. This was my home, and I still consider it my home. I've had many great times here, particularly with Neil and Esteban and many others as well. So it's just fantastic to be here. So I'm going to talk about some work that I've been doing with my PhD student who's almost ready to graduate. I think she'll graduate next quarter, on doing transcriptome-wide association studies in diverse populations. I can talk about some of the challenges, as well as new opportunities and potentially new findings in this area. Please feel free to stop me while I'm in the middle of my presentation. If there are any questions, I really welcome that. But I'm really glad to be here. All right. So to date, millions of genomes have been included in genetic association studies, genetic sequencing studies, to identify, genetic variants that are associated with a variety of human health outcomes and diseases. Many of you guys have been at the forefront of this. And so with genome-wide association studies, you typically have some phenotypes, some clinical outcomes, some traits, and then you have, genetic data across the genome, either or sequencing data or genotype array data. And really the goal is to, identify variants that may be involved in that. So in this situation, you're going to have individuals who have both phenotype data and genotype data. Okay, and so those individuals who have both of those types of data, we try to link the two and try to find variants that are associated. And then, of course, we have to do a lot more analyses to really do fine mapping and really try to find the causal variant. But identifying causal variants through GWAS, as well as whole genome sequencing studies, continues to be a challenge. Even with improved annotation and fine mapping resources. And so one of the biggest challenges is that many times we'll find variants associated with our trait, but they're in non-coding regions of the genome. And many times they can also be in intergenic regions. So of course, we're doing association studies. We're identifying variants that are associated with the trait, but not necessarily a causal variant. So, it could be an LD. With the true causal variant that is generally unknown. So typically you see these Manhattan plots. There's a lot of variants in a particular peak. You hope that the variant that is the highest significance in your peak is driving it, but many times this variant is not in a gene. So there's a lot of times it's not annotation information, and so this continues to be a challenge. Variants that have been identified in GWAS have been enriched for expression quantitative loci. So many times these variants that were identified in GWAS, they're overrepresented in the eQTLs. So basically, these findings suggest that many GWAS variants that are influencing disease through gene regulatory changing as opposed to, to particularly like changing the protein, but so basically changing the amount or timing of the protein that's produced. So eQTLs are often enriched in our GWAS findings. And so this realization has prompted the development of a variety of approaches, integrated approaches to characterize the function of GWAS findings that can be attributed to transcriptome regulation. Okay. We should first talk about eQTLs because when we're talking about transcriptomes, we've got to talk about those variants that are involved in regulating the amount of the protein that's being produced. Okay, and so for eQTLs, we want to find those variants that are involved with gene expression. And so in this case, we look for those individuals or samples that have RNA-seq data or gene expression data and genotype data, and we want to find those variants that are involved in gene expression. And so, of course, you have to have the right tissue types, and so eQTLs, certain tissues, certain variants may be involved in expression, but other variants, they're not involved in expression for certain tissues. So it gets really, really complicated. But most GWAS don't have transcriptomic data on studied subjects, and so even if they did, it may not actually be it may be in blood, and you might need gene expression values in other tissue that you may not have. So gene expression values for study subjects are often predicted using reference transcriptomic data. Okay. So essentially, we have like GWAS data and we have phenotype data, but we don't have gene expression data. So what's often done is that we have some reference panel like at GTEx, where we basically build models to predict gene expression on genotype data. So we have gene expression data, we have genotype data, we build our models to predict gene expression. And once we have the weights and we have eQTLs, or we have variants that are associated or linked to eQTLs to build our model. Once we have our weights for the variants, then we can take these weights and apply it to our GWAS data set, for which we don't have eQTLs and we don't have expression data on. And we impute and get predicted gene expression. So even though we don't have measurements of gene expression on these subjects, we use predicted gene expression values based upon SNP data or sequencing data. And those weights are used to give predicted gene expression for individuals in our GWAS data sets. And then we look for an association between predicted gene expression and our phenotype of interest. So GWAS, we're looking for an association between our phenotype and our directly genotype variants, or genotype or sequence variants, or some summary of the genotypes. For example, burden test, where you're grouping variants. Where TWAS, we're generally looking at an association between our phenotype Y and predicted gene expression, where the prediction is based upon the SNPs and the weights that we get from our prediction models, where we're training based upon our subset of samples from GTEx, where we have genotype data and gene expression data. So I hope that's clear. If that's not clear, please stop me. Okay. So similar, we're testing for genes. These are gene level tests, but we're doing predicted gene expression based upon models that were trained using reference panel that had genotype data and gene expression measurements. Okay. So I think that similar to how we're doing like polygenic risk scores, except we're just limiting it to a gene, okay, a small region, as opposed to looking up all the variants across the genome and putting them all in a model. We're just looking at variants particularly within a particular gene, and then trying to find the eQTLs that best predict that. gene expression. And then once we have predicted gene expression based on these weights, for all of our GWAS samples, we now have imputed gene expression values, and we test for an association between our phenotype and gene expression. Okay. So at the bottom, I have a transcriptome-wide association study analyzes association between our phenotype Y with predicted gene expression instead of testing for gene genotypes. So there's been a number of methods and articles written on how to do integrative approaches to do large-scale transcriptome-wide associated studies, but almost all of them are based upon predicted gene expression. Okay, so these are just a few of the papers that are out there that are looking for association between gene expression and traits of interest. So TWAS has emerged as an important genetic analysis tool for deciphering the effects of genetically regulated expression on a phenotype. Now, a key component of transcriptome-wide association studies is the prediction of gene expression from the eQTLs. So I want to talk a little bit about these prediction models, if you don't mind. So I'm a card-carrying statistician, so I have to put an equation in my talks. So just bear with me for a little bit. So let's consider a sample within individuals when we have gene expression values and genotype values in the gene. And here we're going to let Y denote the vector gene expression values, and let X denote a matrix of genotypes for SNPs in the gene. And really what we want is, for our prediction model, we need the betas. We need the weights that best predict our gene expression values, because we're going to use these betas and for predicting gene expression in our target data sets. Okay, so what these methods are typically doing, they're trying to find the best weights that best explain the gene expression values. So we have X as the genotype values. We don't know the betas. We're really trying to find the eQTLs. And so, if the number of variants you have is small relative to your sample size, then standard regression methods can be used, such as least squares. We could just look, everyone knows simple linear regression or multivariate regression. We just find the beta that minimizes this square distance between our observed gene expression value and our predicted gene expression. So you can find that beta. But it's often the case that, the samples for which we have gene expression values on is often quite small, okay, and in particular for certain tissue. And it's definitely going to be small relative to the number of variants that are in a gene that are going to be considered for prediction. And so in that case, when the number of predictors, number of variants, is much larger than the number of sample individuals, you can't use least squares. And so there are other approaches that people are using to try to find the best SNPs to use for predicting gene expression and to get the betas that best predicted. And so to do this, you have to use some type of penalized regression methods that are quite popular, such as lasso and elastic net. And essentially, it uses a similar least squares criteria, but then it adds a penalty with an L1 norm, so a penalty term, right? Because this basically forces, for example, if you're going to add a variant in the prediction model, it really has to improve the prediction because there's a penalty. So you're trying to minimize this equation here, okay? So, and this penalty term actually encourages sparsity, where many of the variants that are in the model will get a beta of 0. Okay, because this penalty term actually forces many of the variants that are in your model to not have any weight in the prediction, because if you're going to add it, it really has to be worth the penalty. Okay, so this is what is typically used. Okay. So any questions about that? So basically what we're trying to do, we might have thousands of SNPs, there's thousands of variants. We need to reduce the number of variants that we're going to be using in our prediction. So we have to have some type of penalty to basically make the data sparse. So we just have a small subset of SNPs that are going to be using in our prediction model, as opposed to using all of the SNPs. for example, which would not be useful because most of these SNPs are not going to have a role in gene expression. So a popular approach to do this is PredictScan. And so that's a popular method for transcriptome-wide association studies. And so it actually uses an elastic net approach, which is a penalized regression method similar to lasso, but it has actually two penalty terms. And so PredictScan has provided a number of weights. We're trying to get these betas, a number of weights for predicted gene expression from genotyping array or sequencing data. And so they developed this prediction values for these SNPs for genes across the genome. And they used the European ancestry subjects from the depression genes and networks cohort. They use whole blood samples, 922, all European ancestry, and they also use GTEx as well. So GTEx, they have 48 tissues. And so essentially what they did, this process that I just talked about here, they found the betas and they found the eQTLs that best predict gene expression so that individuals can do GWAS and can take the GWAS data and do transcriptome-wide association studies using predicted gene expression, using the weights, the betas from PredictScan. Okay, but the main point here I want to point out is that these weights that were identified and are being used were largely based upon European ancestry subjects. Okay. So Anya, one of her first projects with me, we wanted to see how well does PredictScan perform in predicting gene expression when you apply it across other populations, particularly African descent populations. And so what we did was we took the Javadi's data set, this genetic European variation disease RNA sequencing project, and we wanted to compare the prediction accuracy of PredictScan from PredictScan for predicting gene expression with actual gene expression when we evaluated in the CEU, the Great Britain, the Finland, the TSI, the European ancestry population, just Italians. And we also had in this data set gene expression values from the Yoruban. This is the Nigerian group that's part of 1,000 genomes. So we have RNA sequencing data for lymphoblastoid cell lines. We have gene expression values for over 23,000 genes. And what we did is we just predicted gene expression with PredictScan using whole genome sequencing data and really try to compare predicted gene expression from that we have in our data and compare it with the actual gene expression from their Javadi's data set. And I guess shouldn't be a surprise to anybody is that, you know, here in the purple are the Yoruban, the West African population. And this is basically the correlation. Okay, so of all the genes that we compared, and you see that it does worse in the African population, which isn't surprising because the PredictScan model was trained using European ancestry populations. And so, of course, you know, their eQTLs identified to Europeans may not be the same, and also are not the same in other populations. And so this really just showed that this is problematic if you're trying to use the PredictScan weights and applying it for gene expression prediction in other populations besides European populations. But we also saw some differences as well within the European population, but the biggest difference is from the European ancestry populations to that of the African population.
So Tim, if I'm just reading this correctly, it looks like it drops by half or more than half on average in terms of gene expression prediction.
Yeah, so this is kind of on average here. So basically, I can't remember the details, but it's highly significant.
Yeah.
But it depends on the, you know, I should point out, some genes, it works well across all populations, okay? Other genes, you see huge differences. And so this just says of all the genes looking at it, on average, you see a big drop. But I should point out that there are some genes for which it does well, other genes, it does very poorly. But overall, it's clear that the prediction is actually much worse in the African populations compared to the.
I mean, it's interesting that the picture kind of looks like how much the PRS often fails, on average as well. It's sort of very reminiscent of that.
Yeah, and that's why I would say it's really similar to polygenic risk score, except we're just predicting within a gene. Instead of the outcome being some clinical phenotype, the outcome here is just gene expression. And instead of looking at variance from GWAS variance across the genome, we're just looking for prediction within a gene, but very similar.
And the y-axis is the average correlation coefficient.
Yeah, exactly. It's the correlation. Yeah.
So
yeah, and
between the real expression and the imputed expression.
Exactly.
And so average correlation is relatively low.
Yeah, so it can be. Yeah, exactly. So
for everyone, I mean.
Exactly. It's not great. It's great, right? Because but yeah, it's not great. But for some genes, it does very, very well. And I should point out here that in many cases, and reason why we did R instead of R squared, because we wanted to point out that in many cases we found that the correlation was opposite. Like it was a negative correlation, where the prediction was in the opposite direction in the African population more than you expect when using PredictScan. Yeah. But but there's a lot of factors that influence gene expression. And if you look at the gene expression prediction values, they're not great. A lot of them actually, are around 0. But there are some genes where it is the prediction is quite good. Here is a paper.
Sorry, have you looked if there's anything about those genes that could explain that? Are there like very commonly, expressing in all different cells or?
Yeah, so those ones that do really great is that typically there may be only a few eQTLs and we identify them and they really drive the expression. So the effect sizes are really, really large. Yeah. There's a paper published last year that really showed that similar what we showed is that when predicting using ElasticNet models using European only and trying to predict it in your Rubin, you see the prediction is actually quite poor relative to what you would expect. Okay. Here was a paper that came out of UCSF, led by Kevin Keys and Chris Genois and Esteban here. And really they explored quite nicely, the power of TWAS and just really showed that how much power is actually lost when your training sample doesn't match your testing sample. And this was the case if you trained your prediction models in African and try to predict in Europeans, you saw the same problems as if you trained in Europeans and try to apply it to Africans. And so really nicely complemented the work that we previously did, just showing what the problems were that PredictScan doesn't do very, very well in predicting gene expression. And then here is actually taking the next step was once you have gene expression values, now you want to use that in the TWAS and just showing that how the power is actually diminished when you don't have good gene expression prediction in one population. And so basically, and it's really consistent. It's interesting too that the African Americans, for example, fall, like if you train in African ancestry populations and you apply to African Americans, they're somewhere in between the Africans and the Europeans, which makes sense because they are at admixed. And so you can see, for example, the African Americans fall somewhere in between, the African group and the pure European group. But it's not surprising, but it really dove quite deep into really understanding how the power changes if, for example, if if the populations don't share any eQTLs, okay, or if they share half the eQTLs, or if they share all the eQTLs. But even if the same eQTLs are even if the eQTLs are the same in both populations, the African and European, still there is still a loss of power in that case as well, even when the eQTLs are the same. And one of the reasons for this is that, a priori, these machine learning methods, these penalized regression methods may not get the true eQTLs. Okay, and so if it doesn't get the QETLs, it tries to find the best combination. And so many times, because different LD across populations, it may not get the true eQTLs. And so in this case, you still have some loss in power. So Anya's second project was to see if we could kind of improve prediction of transcriptomics in diverse populations. And so she came up with a beautiful approach to this, and it's called a fused lasso method. So basically to improve gene expression. So what it does is we have two penalty terms. So I introduced the lasso before. So let's say we have K ancestry groups, and we know which group these individuals should be a part of. A priori. So we can do this, for example, based upon PCA, and we can cluster individuals or self-reported ethnicity or ancestry. However, we want to cluster individuals, but let's say in this case, simple cases, we have Africans and Europeans, or we can also have more. And so what it does is it allows every group to have their own betas, their own weights in the prediction model, but it forces those SNPs for which the weight should be the same. They have the same eQTLs, effect size are the same. It tries to encourages population to have similar weights, but when similar weights don't fit the model, it allows the population to have different weights. So basically, it tries to get the best of both worlds. When the eQTLs are the same, it tries to give the similar weights and tries to identify the same eQTLs across populations. When they are different, when the eQTLs appear to be different, it allows each of the groups to have different weights in the model itself. And so basically, the L1 penalty basically tries to encourage sparsity, right? Because we want a small number of variants in the prediction model as possible that will best explain the gene expression. And then we also want to force the coefficients to be similar if indeed the eQTLs are the same. But if they're different, then we want to allow for them to be different. And we looked at this under different genetic architectures. So for example, there's a purple population, let's say that's the African population, and the yellow is the European population. So we looked at the case when the eQTLs were the same, in the same direction, in the same effect size. And so fused lasso is our method, and then these are like the other methods, but fused lasso in this case does quite well. I should say that in most cases, the optimal situation is when you have the same reference sample, the same training sample is the same as your target sample. But if we don't have that information, or we can't do that, this is a way for us to kind of that works quite well in a lot of different situations. So here's a situation where the genetic architecture is the same in both populations. It's quite well across simulation studies. I won't go into too much detail because I have a lot to cover. Here's a case where in the same gene, for one of the eQTLs is the same for both populations, but but the second eQTL is different. And here is a situation where we have different eQTLs, but they're still in the same direction. And so our fused lasso here, sorry if you can't see it so well, does pretty well. So this is when this is like the ideal situation where you actually have the correct reference samples for everybody. But in all cases, our fused lasso seems to be quite robust. And here's a situation where we can have different effect sizes. So the eQTLs are the same, but they have different, the effect is in a different direction. And in that case, we actually do quite well in that situation. It's still quite robust. And then the final is when the eQTLs are the same, but they're in different direction. So there's still more work. We're writing this paper up, but this fused lasso approach seems to be quite robust to the underlying genetic architecture of the eQTLs, which often the case is going to be unknown.
Tim, sorry to interrupt. I think
Sure.
Linda had a question, and there were a couple of questions I could read from the chat, but
Sure.
do a second.
Sure. Thanks. Well, I think you partly answered my question, but so with this method, it seems to me like one issue that it's trying to overcome are basically deficits in performance because of LD differences between populations. So this sort of seems to be able to tune the weights a little bit more to LD differences, right?
Yeah, exactly. But not just LD differences. What about if effect sizes are different and in different directions as well?
Yeah, that was quite interesting. Did you find a lot of instances of like SNPs being eQTLs with opposing directions of effect? Like that's really interesting.
Yeah. So this is I should point out that this is simulated data. So we were evaluating. So I'm not showing. So we actually applied this to TopMed and just we looked at for the MESA data, and we did see an improvement as far as predicting. And so we haven't gone back to look at or categorize what proportion of the gene expression variants that were selected are in the same direction or different. But I'm glad you brought that up. I think that's something we should actually look at as well. I think that'd be important.
But it's still nice to see that the method has flexibility and is able to handle that.
Yeah, it seems to be robust. But I would say, you know, the best, if you can get a good reference sample to match your target sample, that's going to be ideal. But like I said, but there are cases, for example, if the direction is the same, if the variants, the same eQTLs are the same, there is some advantage of using all of the data and not separating it. Okay, so by a priori, we often don't know the underlying genetic architecture of the eQTLs.
So also, Tim, from the chat, there are questions from Neil and Mark Southstead. So the questions were, is this, I think it's the poor performance. Do you think it's due to allele frequency differences, LD, or both?
Yeah, it can be both. I mean, so we looked at both. So we looked at if the allele frequencies are different. So if the allele frequencies are different, then the effect sizes are going to be different as well. So I should point that out. But here, for example, it can be to the betas. But what we also looked at was really interesting is because of the LD being different, what variants get chosen by these machine learning methods, it differs. It doesn't actually pick the correct eQTL all the time. Many times it misses it, but it can still do quite well if it is able to get variants that are tagging the eQTL. And that's where the LD makes a difference. So yes, so it is the frequency of the variants, but LD is important, but also the genetic architecture. Are the eQTLs the same? And so, and another point that I actually haven't pointed out yet here is that we do see significant differences for many genes and differences in gene expression across populations as well. And I think Esteban had a paper recently about that, saying that yeah, many genes have expression levels that differ by ancestry or ethnicity. And so Esteban can talk more about that than I do, but I'm assuming that might mean that they're potentially different eQTLs or different effect sizes. I just don't know, but I think it's very, very complicated.
It's led by, that work is led by Angel Mock and Linda Kachuri, who are on the phone, and Elad, so they could chime in.
Okay, please do.
I have a comment. I put it in the chat, but this is all very reminiscent. I think you said this is all very reminiscent also of what happens with GWAS. So in our experience with the GWAS, what happens, and I guess, again, with the GWAS, it's just because the reference samples always are largely European, as everybody knows. And what happens is that because of statistical significance, you get the allele frequencies you get are closer to 0.5 or away from 0 and 1 in the Europeans, but they tend to be more extreme towards 0 or 1 in the other ethnic groups, including Asians, as well as Africans and African Americans. So fundamental, and that has a large, in our experience, had a lot to do with why the variance explained is lower in some populations. It's just because those variants are not polymorphic in other populations. And that just has to do with the discovery, because the discoveries focus on a different ethnic group. But you're raising a very important fundamental question, because we've also looked at effect size differences.
Mm-hmm.
And, and that's an important distinction to make, as you're bringing up. It's one thing that, for differences to occur because of allele frequency differences. It's another if you have exactly the same variant that shows a difference of effect, in the different ancestral groups. And there's potential multiple reasons that could happen, but that's important in biological distinction, because why would the same, and I think as Mark pointed out, it could be that because neither of the variants or one of the variants is actually not causal, that it's an LD issue, but when you say something is an eQTL in one population, but it's not in another, and another one has a different one, that would be my question. Are we talking about the fact that it's not polymorphic in one population, therefore it's not an eQTL in a population, or that it exists in the other population, but the beta coefficient is significantly different? And explanations for that. So that's, I guess what I'm trying to think about here, what's the biology underlying this? And I think, especially for a reversal, I think Laura was asking about this too. Those are pretty exceptional, I think, when you'd have an effect going in one direction, one population, and a different effect in the other. One other comment I just make quickly is that I imagine, the context is important, and that's why the different tissues, that's why people are always deriving these eQTLs from different tissues and so on. But also, they do these expression studies in people who are on drugs or people who are, have different kind of environmental exposures, because the expression is not just a consequence of the genome, it's a consequence of the environment, right?
Yeah.
I imagine. And so there could be significant differences. And, so some eQTLs may exist in one environment, but not another. And so even when you find an ethnic difference, you might have to consider that the environment in which those people lived, could be driving expression differences also, that it's not necessarily just genomic, right?
Yeah, but Niels, but I thought we're largely just capturing the genetic component that's contributing to gene expression, right?
But that's from GWAS on expression data, right?
That's right.
So
yeah.
That's based on a GWAS study of expression data, and you're using a 5 times 10 to the minus 8 whatever threshold.
That's
and again,
right.
that has to do with the discovery side. But remember, just think about, it's a GWAS analysis of expression data. And so what was the source of the expression data, and
That's
whom,
right.
and what was the environment in which it was created? So this is, I'm not being, this is all great, actually, what you're doing.
No, that's a good point.
But I'm just trying to get to some biological explanations here also of what might be creating differences.
So how about multiple eQTLs that affect expression of a gene, and you might have one that is more frequent in one population and less in the other. Now you have these two different frequency SNPs that affect over, and then it looks like the beta is different, that the effect size is different, but it's really related to the frequency of the different eQTLs in the gene.
Yeah, I think that's so I think,
Sorry if we derailed your talk, Tim. I'm sorry.
No. This is great, because like I said, we're struggling with this as well, because I guess what we're thinking is that we're not getting the true eQTL that's driving, right? I'm not even certain we have the correct eQTLs for the Europeans. I think we have variants that are tagging the true eQTLs.
Well, now you're back to the fine mapping question you brought up at the very beginning.
Exactly.
And how do you do it? So there's the question. So usually they do LD, usually they'll go to an African population, because there's less LD in the Africans, you get better resolution of the LD, right?
Yeah.
I don't know. Are you doing that? I mean, are you proposing to do that for expression data? Maybe, the expression data to me is not so different from any phenotype because it's all based on GWAS, right?
That's right.
So the principles should apply. So you could argue, I would think, that for a much more diverse sample for doing expression studies, and especially focusing on Africans who have less LD, to help to try to resolve what's causal.
Yeah. No, I'm with you. The one example that's coming to my mind, and I know this came out of Peggy Vance's, and that like the example is like ApoE. Maybe there's something special where they're showing that, like, we know the effect of ApoE, 4 variant is different and is highly diminished in Africans versus Europeans, but they're also showing, and so this is a true causal variant, but also expression levels are different for even though they have this, comparing Europeans who have the E4 compared to Africans of the E4.
Well, didn't you had a brilliant paper? Didn't you show that the haplotype, the haplotype on which the allele exists also mattered?
Yes. So, and that's what I'm saying. So there's more going on in that region than just the ApoE. Yes. And I think that potentially also is driving different effect sizes.
Right, and that could be the interaction between different eQTLs for the same gene that I was mentioning.
Well, right? And it's not just local, it could be global genomically. It could be global. The genomic, when I say background, there could be environmental background, but it could be genetic background also is interacting, which is creating a difference in the betas also. So, but again, I think it's important when you talk about variance explained or whatever, to try to dissect apart how much of it is due to allele frequency differences of those variants and how much of it is actually due to effect size differences. And the effect size differences could be caused by a number of things which we've been talking about.
Yeah, I agree. Very nice.
Okay, let's let you talk.
Okay,
to the next slide.
How much time do I have left?
You have 15 minutes-ish.
15 minutes? Oh my goodness. Okay, let me move.
We'll stick around longer.
Okay, that's fine. Okay, all right. So, all right, so now let's say we have good expression prediction. So, and it's hit or miss, as you'll see. So what we wanted to do, we wanted to do a TWAS in TopMed. So for those who don't know, TopMed is a transomic precision medicine program. Whole genome sequencing data is available on about 150,000 or so individuals. Many of you are involved in TopMed. I know Esteban's samples from Gala are part of TopMed. But there are about 51 ancestrally diverse cohorts with well-defined phenotypes and existing clinical outcomes data available. And so associate analysis are still going on with whole genome sequencing data for a large variety of traits. Some of these papers have found some novel discoveries. And so, we focus on, at least for a few years, developing linear mixed models for single variant testing, but to account for the complex sample structure in TopMed. So just a figure, just showing, for example, how complex the Ancestry structure is. So we develop a method to kind of how do you infer, population structure in the presence of relatedness. And so here, for example, just this is the first 2 PCs, and this is just showing how diverse the TopMed samples are. And the parallel coordinates plot is really nice because it allows you to plot multiple dimensions of PCs. So a single individual, you just follow them. That PC1, this is their value, and PC2, and just follow them along the coordinates. So really just shows you that there's a lot of heterogeneity in ancestry, and the PCs are largely capturing that, but quite diverse. There's a lot of relatedness here. So we had a method to infer relatedness in the present population structure. Actually, I got into this area because of Neil, because we were looking at the Women's Health Initiative, African American, Hispanic, and found that we couldn't identify relatives using standard approaches. And Neil and I worked on this, and we found that we need to use individual specific allele frequencies based on ancestry proportions to deconvolute relatedness from population structure. And then I follow this up with my first PhD student where we don't actually have to need the ancestry proportion, and we can use PCs, but it performs similarly. But this is showing like there's a lot of relatedness here. On the x-axis, I think is the IBD0 sharing, and on the y-axis is like the kinship coefficient or something like that. But there's a lot of relatives here. So just know there's both population structure and relatedness. So we develop a whole bunch of methods to kind of account for that in TopMed. I'm going to use as example white blood cell count. So Anya and Paul They led analysis for TopMed for white blood cell count. We had Amish, Asian, Black, Hispanic, and white. And then they did a GWAS, large-scale GWAS, fixed effects, adjusted for age, sex, the study, the phase, top PCs, random effects for relatedness, inverse, normal, transform the white blood cell count, allowing us to kind of analyze all the different cohorts to get together. And this is just the GWAS results. And so found 7 newly discovered association with white blood cell count. And this was recently published in American Journal. So this is what we found with white blood cell count. So if you're interested, please have a look. I think it's a really good paper, and really shows the power of combining diverse samples for novel discoveries. So the linear mix model that we used, accounts for all that structure. And then what I asked Anya to do for her third project is to let's use our predicted gene expression and let's do a linear mix model for TWAS. So instead of testing for a variant for association, we actually use imputed gene expression. So here we're looking for an association between our phenotype, white blood cell count in this example, and predicted gene expression. So we want to test whether or not beta 1 is equal to 0, but we can account for all the different structure in the data. So it's very similar to the linear mix model for GWAS, except now we're using a linear mix model for TWAS, and we're using predicted gene expression. I think what's new here is that we're doing a mixed model, allowing for a complex sample structure, but it's very similar. We also allow for differences in not just the mean differences in gene expression by ethnic groups. We also allow for the variance to be different, because we also found that for some of these gene expression values, the variance also differed by race and ethnicity. We always focus on the means being different, but you got to account for the variances being different as well. So we actually included another component that allowed each ethnic or genetic or ancestry groups to have their own variance as in addition to having their own mean. Okay, so we did that, and so we also included some computation efficiency improvements by using sparse matrices for kinship. I won't go into detail here because we don't have a lot of time. And so we use PredictScan. We use predicted gene expression using whole blood tissue, and using TopMed whole genome sequencing data for gene expression. So we got about 12,612 genes that were predicted. So in our whole blood sequence data. So a lot of genes, for example, may not be turned on or don't have good prediction values but for PredictScan for whole blood, there were 12,612 genes that we could impute gene expression.
Wait, what's the I'm lost about the reference now for imputing the gene expression.
Yeah, so this is using PredictScan. So we're using PredictScan. So these are, I think they're using
so it's the non-diverse.
Yeah, exactly. Yes, exactly. So now what I've heard is that they're supposed to have a new prediction that is more diverse. And I don't know if it's a PredictScan, but I know there are new, like MESA is having their own prediction based upon race and for different ethnic groups. But in our case, we just use the gene expression from PredictScan for what I'm showing here. So, and I should say that as I mentioned, I'll go back here. Even here, I'm sorry, I'm all over the place. Okay, so here, you could still, even if the model is not, even if you're using a different training set than you should, you can still get some signal here, right? Even if the training sample is different than the test sample, different ancestry, there can be some signal because there are some overlapping eQTLs. Okay, but optimal is going to be your training sample having the same ancestry as your test sample. So we applied this to first MESA, because MESA we have the gene expression measured, and we looked at predicted gene expression, and MESA is quite diverse. And just so you know, like the median is about 0.006, the mean is 0.032. This is the R squared, okay? So for many of them, it does not predict very well, but there's some where it predicts very, very well. So at this line here is the median, but there's some where the prediction is quite good, okay? So we ran our TWAS and we're quite surprised. So we found 97 genes with the predicted gene expressions that were significantly associated with white blood cell count after bone and foreign correction using the same samples that were used for the GWAS. But now we're getting gene expression levels. So you see, we're seeing 97 genes that were significantly associated after bone foreign correction. So these are all the genes. Many of them overlap. So we looked at the GWAS catalog. There were 143 GWAS variants included in the catalog. 29.9% of those GWAS variants were actually in intergenic regions. So an advantage of this is we are finding associations with the gene itself, okay, as opposed to many of these GWAS variants are in intergenic regions. And then we looked at those GWAS variants that have been reported and compared to ours. So 24 of our significant TWAS genes were previously identified. And so they do match the genes that were reported in the GWAS catalog. So these are the ones that are that overlap. And then these are the novel genes that were identified by the TWAS. Now, I should point out, I suspect many of these genes expressions are highly correlated with each other. And so, our next step, we have to figure out how many of these genes are independent. And don't forget, these are predicted gene expression values, right? So they're using the SNPs. So the same SNPs that are being used in this gene, for example, there could be some overlap with those same SNPs in some of these other genes. Okay, so because the predicted gene expression values is based upon the GWAS variants or the variants that were tagged that were identified to be eQTLs. And so so who knows, the overlap, but I suspect there probably is a lot of overlap. But here is the TWAS and the GWAS results. And so you see that there is a pretty good correspondence between our GWAS results and the TWAS variants. But the advantage of the TWAS in this case, I would say, is that we can talk about the gene expression and we know the gene, whereas TWAS, many of these variants are in intergenic regions and we don't know what role it plays. So important here is that the most important variant for white blood cell count in African Americans is the Duffy variant. And PredictScan was not able to impute or provide gene expression for the Duffy variant because they build their model on European ancestry training data. So I suspect that there's many variants that are not being used, or many genes that were not, that may be expressed, but that aren't going to be provided by PredictScan. And like I said, to me, this was a glowing problem because the Duffy gene is the most significant gene for white blood cell count in African Americans. And so the fact that for this particular trait, PredictScan couldn't provide that really highlighted the importance of more diverse training samples for improved prediction of transcriptomics in diverse populations. So we're still digging into these results, trying to figure out what's actually novel, how many of these genes are actually independent. So, we're going to do the whole process of, conditional analysis and things of that nature, and trying to figure out, how many of these genes actually are independent. But I was really encouraged just to see these results because it is providing something complementary as well as different from the GWAS results. So in closing, TWAS can be done in large-scale multi-ethnic cohort analysis. They should account for relatedness, population structure. As I just mentioned, TWAS can give complementary as well as different results. And I think that if we can have improved prediction models for transcriptomics, I think there's a lot more that we will be able to uncover. And so with that, I'll stop, and I'm happy to entertain any questions and discussion.
Thank you. That was outstanding. I guess I have a interesting interest in Duffy personally. I guess it sounds, it's a good idea probably to condition. The only other thing that's interesting about Duffy is, I guess, and I don't know how many of these, how many variants there are like this, but the Duffy works particularly well in terms of probably predicting, gene expression and white blood cell count, also so on, in admixed populations, right? Or in populations, or let's say in Middle Eastern populations, North African populations, East African populations, where it's more polymorphic. I probably not a lot of examples like this, but it also does bring up the fact that admixture has occasionally had some unique properties to bring out some effects.
Yeah, I suspect that once we condition on Duffy, but we didn't have the Duffy gene to condition on, but I think we're going to just condition on the Duffy variants. I suspect, I mean, it's got to be driving these results. I mean, it has to. It's just hard for me to see that all of these, like these are really novel genes. I mean, because these are supposed to be novel, but I just suspect that Duffy is driving that.
I said that, actually, I said that in the chat. Also.
Okay, I can't.
Yeah. Because it's like all the genes, all these expression QTLs are in the same place on chromosome 1. Yeah. So no, it's got to be Duffy.
Okay.
I made another suggestion. What happens if you take out everybody who's got any African ancestry from the analysis? What are you left with? Because then you're going to get rid of Duffy. Okay. So then what are you left with? And maybe you're left with what's on the other chromosomes here. And you know what? And the other thing that would be of interest also, this is the Duffy obviously is a very strong effect here, but looking for interactions, in the expression levels or whatever with these other loci, with these other locations. And how many of these are driven like on chromosome 6? I don't know. That looks a little HLA-like? I don't know.
Yeah.
I don't know if that's HLA. Who knows what you're going to get, because HLA is like so polymorphic, right?
Yeah, HLA is in here. I don't know if you probably can't.
It is. I saw it there, and
it's there. But
oh my gosh! Good luck. That's all I can say. Good luck with that.
Yeah. No,
HLA is so polymorphic. I don't know how you're going to resolve any of that.
Exactly.
But it's there. There's clearly an effect.
Yeah. So this isn't my area of expertise, but you tell me, those who are experts in the area, like all we're getting is expression values, but it doesn't tell us the protein, right? Or that's being expressed, right? Because you just talk about HLA being quite polymorphic or different, but I mean, we get expression levels, but we don't know the difference in the protein.
But the expression level is based on a gene. So
yeah.
So presumably it would be the expression of the protein of that gene.
Yeah, but the protein can be different, right, at a gene.
I don't know.
Is that right? I mean, those who are like,
I don't know. I'm assuming.
Are you asking whether there are sort of non-synonymous variants that also could be part of the haplotypes that are associated with expression? Is that, in other words, like there are coding sequence variants that are potentially being tagged by the expression variants?
So wait a second. It's based on the expression, the level of RNA. Of a specific RNA, right?
Yes.
So you're measuring the level of that RNA sequence. So the RNA, I presume, is coding for a protein. It could be expression of any RNA, but I'm assuming that's what it is, right?
It could be like you're combining isoforms, but I don't know how would you get into distinguishing that.
Yeah, that's what I'm
wondering. So actually, I mean, you could. There's been. I mean, you could look at splicing, and so there have been SQTL sort of TWAS extensions of TWAS that look at different splicing QTLs, and so then you can look at different gene isoforms. But I think to your point about the proteins, I mean, I do think there's also value in integrating TWAS with something with like basically rerunning the same analysis using pQTLs, protein quantitative trait loci. And I think this is sort of maybe. I mean, HLA is a bit of a different situation because there you have to get into like specific like functional alleles, and I think that's unique. But just for other genes, I do feel like sort of doing like a TWAS and a PWAS, I think would be.
What data is available to do PQT protein? So you need to have protein level data for a lot of people with GWAS.
There's some reference data for this. There's like
And,
an EQTL consortium.
I think there might be
Same
one
issue
other
with
thing.
Ancestry though, unfortunately, I think.
I think there may be one other thing you need to pay attention to. I think, I'm maybe not the expert here, but the same variant could influence the expression of more than one local gene, one local protein. So my question is also, if you have like neighboring proteins or transcripts or whatever, how correlated are their expression levels? And those may be influenced by the same variants. So like on chromosome, that's another question I have. When you're finding, a lot of different genes in the same region, underlying, coming out in your TWAS analysis, I'm just wondering, are they co-regulated? Is this just due to LD, or is this just due to one SNP, or what?
Yeah.
This is where you need to do experiments, right? Where you can do like editing and start looking at what happens.
Or just look at the correlation structure of local, transcripts.
Yeah.
And I don't know how much, people have done that. I presume they have.
Yeah, I
Yeah,
think there are
that's
methods that have been developed to kind of like quote unquote find map a TWAS, because I think it is sort of, it is hairy sometimes when you get a result like multiple correlated genes, Neil, as you said.
Yeah.
And-
But and don't some of these genes they overlap with each other. So presumably they should be sharing.
Well, you can have anything. You can have genes within a gene too.
That's right. That's right. Yeah.
You can have anything. Right.
No, and I think that this
Yeah,
is
welcome.
this is to Lara's point, though, that maybe, at the end of the day, association testing gets us to a certain point,
Yeah.
and then the correlation structure is such that we can't resolve it anymore.
Exactly.
And then, have to call up our functional, experimental colleagues and
Agree.
say, hey, if the gene, the trait is important, the gene is important, it's worthwhile probably pursuing some functional work.
Yeah. So, okay, great. Well, thanks.
Can I ask
so much
one more
for
question?
the questions? Yeah, sure, Neil.
So you just showed data where, was it Mesa, whatever, did transcripts on 1,000. Why would you be using the original predict scan when you have so many more subjects on whom you have expression data that you could use as a reference? Or maybe I'm just confused about that.
Yeah, so there are 838 subjects, but it's really diverse. So they have African, they have Europeans, they do have African American Europeans, they have Hispanics. So it is quite diverse, but they have created their own prediction models as well.
Yeah, so why could you use those for the rest of TopMed? That's what I'm asking. Usually like when we do for imputing genotypes, you want to get a good size reference as diverse or whatever. Wouldn't you want?
Yeah,
but it's what I said before. Don't you want more Africans or African Americans or something in this?
Yeah, we would. I agree. The limitation is that they only have it for blood.
Right. So So how important is the tissue context?
Yes. Yeah. Exactly.
Right.
Yeah. So that's the only limitation. But I think for like blood related, tissue types or whatever that might be of interest, I think this will be ideal. But for other tissue types, I don't think they don't have that. I know. So Esteban's on here. So is Esteban still here? I can't see because I'm still in full screen mode. But Esteban, I think you're getting some other large scale gene expression values, right, to build these models?
Yes.
Did we lose it?
This is a lot. I'll speak for him if he's not on, but Linda and Angel are who are on the call are kind of running that, and I'll let them say a word.
Well, I guess I'll just sort of say, so we have been working with Esteban's data set that is part of TopMed, and it is a diverse sample, and we have been developing TWAS models for prediction of whole blood gene expression, and in a very diverse group that includes African Americans, Mexican Americans, and Puerto Ricans. So I think that's a resource that I think we would like to make available for the community. We're still working on it, but I think again it'll be blood, so it doesn't really solve the issue of what's happening in other tissues. But yeah. I don't know, Angel, if you want to add.
Great to have. That'd be a great resource.
Actually, I have sort of a question. So I think MESA is also part of TopMed, I believe. Are the models that you're using the ones that have been published a few years ago that are based on the array data, or has this been like an update of MESA with sequencing?
So we've been doing the array.
Okay.
Yeah, so we're using array data. Yeah.
Yeah.
And to Neil's point, we're actually going to go back because right now we use the predict scan. So we use the latest predict scan for whole blood tissue. We really want to go back and swap out those predicted gene expression using the MESA weights. Yeah, but I guess the limitation is, these people, like African Americans, they're admixed. And so, we've been looking at like, should we be doing gene expression based on local ancestry? And so that is, and we tried that and we didn't get a huge improvement. Like I said, but probably
we also tried that and didn't get an improvement.
Yeah, it was so baffling because we spent a couple of years just trying to get it right, and we just realized there's just not much to be gained by using local ancestry, and I don't have a good reason for why that's the case. But like I said, as most of the things don't really predict that well. It's kind of like what Neil was highlighting is that there's so many other factors. We're probably not at the true eQTLs, and there's so many other things that influence gene expression. But we've been using global ancestry and things of that nature and putting them to subgroups, discrete subgroups. But it does seem like we should be able to improve more by taking it into account.
I'm just having a quick thought here. If local ancestry of a particular gene doesn't make any difference, but global ancestry does, that would suggest that it's not just genetic factors that are at that gene that are influencing the expression, but genes around the genome.
Yeah.
Or environment.
Environment too.
Or environment, but I would suspect that it's genome-wide, just like everything else. There's like polygenic effects that, and it's global ancestry that's mattering more than the local ancestry. It sounds like that's what you're saying.
Yeah, exactly. That's what we're finding. Because
that's another very important point.
Yeah.
Because you found the racial differences, right? And
yeah.
The question is, and it's not local. So it appears like that difference is not due to local differences.
Yeah.
Ancestry differences is due to the global ones.
Yeah. Yeah. So, I mean, so what we tried to do, Neil, is, for example, if for African American, that a particular gene, if both of their genes were from a European background, we were using the European model.
Right.
And if both of the genes were from an African background, we were using the African model for predicting, and these just did not work well. And I think it's what you just said. It's like there's so
you should write this up. I mean, really, this is very interesting and it's very important. This is, because it says a lot about, whether the influences on transcription and so on are really locally driven or distally driven. And it sounds like there's a lot of distal driven effects here.
A lot more going on. You're right. Yeah.
I guess in our situation, the way we're sort of thinking about it is it seems like the main driver of the difference of performance is the degree of ancestry mismatch between the training and the testing data. And so I think once it sounds like, you're using a more ancestrally close reference data set on the eQTL side, that I think that part of global ancestry makes the biggest difference. And so this is sort of our hypothesis was that once you account for that, then it's possible. I feel like that maybe local ancestry could be helpful for like fine mapping causal eQTLs, but in just sort of like a broad TWAS setting where you're just building prediction models, it just doesn't really seem to make much of an impact in terms of like overall prediction.
Yeah, but one thing I was thinking about, and maybe this, I'm thinking about like Elizabeth Atkinson's paper recently where, for polygenic risk score, we're using an ancestry specific, effects for, PRS. It seems like this should be possible.
So it's interesting that you sort of bring that up, because I think her method is, I think it's really promising, but I think there's been some like additional work showing that I think it works really well under some very, very specific contexts where you have a lot of effect size heterogeneity and like large effect size heterogeneity. And I think actually in a lot of other contexts, I think there was just a published paper that was published recently. They show that actually Tractor can sometimes have poorer performance and lower power. So I think it could be.
Well, yeah. Tim was also putting in a penalty for differences.
Yeah.
you know, he was penalizing differences, trying to make them more comparable in
That's
terms of
right.
effect size, which I think actually makes sense.
Mhm.
Because again, you're always going to find differences just by randomness,
Yeah.
right?
Exactly.
That's not, you're right. So, which you're going to pay, you're going to pay a price if you're going to allow for that randomness, right?
Yes,
So I think
yeah.
you're absolutely right to do that.
Yeah.
I think your approach, the fuse lasso, I think might actually be more flexible because with Tractor, there's like an extra, degree of freedom that they're using to test some of those differences. But I think it's the more adaptive tuning of the weights to LD. I think that for TWAS, at least, it sounds like that would be very promising.
Yeah. So yeah, and like I said, we like I said, the biggest limitation I say is that we have to put people into discrete groups, which I do not like, particularly for admixed populations. But I just don't have a good alternative because the local ancestry thing just didn't work out for us.
Well, why, I'm not sure why you have to put them in discrete groups.
Because like I said, we're getting betas for each group, right? So we got to get weights. So
Yeah, but theoretically you could calculate a beta as a function of ancestry.
That's true. That is true. So you're saying use the global ancestry, maybe?
Yes, you could.
Okay. That's
Well, I don't know. I think Esteban, Elad, didn't you guys look at global ancestry and expression levels?
Is Esteban? I think we lost Esteban.
Well, I would know. I mean, you guys.
Yeah.
I thought Noah did this. I thought you guys did this.
Noah's done some stuff. I'm not sure I remember all of it, but I think when we looked at it, and actually Linda and Angel actually know the most, but I think when we looked at it, local ancestry had effects, but basically they were kind of subsumed by the variant. Like if you have full sequence data, and you're not hiding variants and stuff, it works better than Ancestry, which I think is, to me, makes relatively straightforward sense. It's not really, there's nothing mystical about the Ancestry. It's just, for the most part, there are different allele frequencies that if you capture in an adequate way with a good sample size, you get good estimates for the effects and they work well. There are occasionally, I think, some genome-wide ancestry effects, which I don't think we've dissected fully.
What if you estimate like the effect modification of ancestry proportion on the beta and then use that?
So that's kind of what Tractor does, I think, if I'm not mistaken. Somebody can correct me, but it does. I think it works well when there's when there's a lot of heterogeneity.
Yeah, but so you can, it can
when
be
isn't
like,
heterogeneity, you pay a price. Because I think it's sort of like,
it can be zero. That effect can be zero for the ones that have no heterogeneity.
Sure, right.
Estimating based on ancestry proportion.
Right. I mean, I think it's sort of like the balance maybe between, Tim, what your method was sort of doing, which is sort of like penalizing for differences and sort of, I think, in a way, the Tractor sort of like under penalizes differences, or sort of assumes that there's always potentially a difference, and kind of opens up another degree of freedom, which then can hurt power when there's not a lot of differences. So maybe a balance.
I think Tractor misses Duffy. Like I think that's kind of an issue.
But was that
no, not Tractor. PredictScan did.
No, Tractor. I think in the follow-up, so people who did follow-up work applying Tractor to different contexts. Oh wow. I think it misses Duffy. So anyways, I mean, having not used Tractor myself.
There's no Duffy in the Europeans? Is that because there's no Duffy in the Europeans?
Sure.
Probably, or I think maybe even I would have to look specifically into the details of like why that happened, but yeah, I think maybe because maybe you can't test for heterogeneity because it's just like not present in the other.
Yeah, that's interesting.
Yeah.
If it's not expressed in the data, there's no data.
Right. I think it's just kind of an example of like why you can't just automatically just apply one method to every population. But just going back to something that you said earlier, I mean, I think it's a big challenge, sort of how we approach doing TWAS and whether at some point you have to pick discrete groups. Like in some of the work that we're doing, we're defining individuals as having predominantly African ancestry, and we're using like the median as the cutoff because we have a very heterogeneous, very admixed sample. But then, you know, when people maybe want to go and apply these models in their data set, if the ancestry proportions are different, then right, there'll be implications for how well our models port over. So I think that's definitely kind of an open question. Like, we understand that we want the training and testing to be as similar as possible, but in practice, how do we actually implement that?
Yeah.
I'm wondering also when the eQTLs are being discovered, even if you have a diverse sample, but the proportion of diverse individuals is very small, if when you're looking at genes, you don't lose, maybe you lose genes during the QC because you're getting rid of things with a lot of variants or very few people expressing, you know, like all the steps you use for you know, ending up with gene counts, read counts, of, you know, whatever you're using as your matrix.
So how about using principal components as your unit?
Yeah, but I think the problem with the PCs are sample dependent, right? So how are we going to apply that to another data set, right? Independent data set, which will, how do we match the PCs from the training set to the
Well,
test
you
set?
can't you just, but you can use the coefficients from the PCs. You can just, like we do this all the time. You know, you generate the PCs from like an ancestral reference population, but then you apply those scores to an independent
project.
sample. You project them. It's like you project the other
That's a good
individuals
idea.
onto the PC figure.
So then everyone has to project from your reference and then
Right.
use the
You have
weight.
to-
Well, if you have a nice big reference that's very diverse, it probably would work okay. As long as you have the same set of SNPs.
That's right. Yes, I agree. Yep, I see where you're going with that, John.
I mean, this question that you've raised, I mean, it's really important because I think we have to start moving away from these broad categories.
Yeah,
So how to do it it's complicated.
exactly. Yeah.
Well, the other thing is, you know, when you're testing a goodness of fit, African Americans, as you know, like can range in terms of African ancestry from 100% to 30% or 20% or whatever. And so you might want to calculate goodness of fit as a function of that ancestry as well, not just as a group.
Well, and the other thing about the PCs, I think, is that they really, you know, they obviously reflect genetic variance and you know, kind of could get at things like, I mean, if you have a European and Asian subsets, for example, and then you know, your individuals actually have, like, you know, there's South Asian, Central Asian, whatever, that kind of categorization, as you've already said, implied, is not ideal. And so, you know, kind of the natural distribution of human populations, sort of the demographic history, is probably more accurately reflected in the PCs.
I agree.
Although they, you know, there's all kinds of other issues with the PCs, like, you know, how many, and, you know, kind of, you know, you take the European subset out and then you know, do PC.
Well, the
again.
PCs are always a function of who you included.
Yeah, exactly.
Yeah.
Like in America, you know, like in San Francisco and Boston and New York, you'll get a component of Ashkenazi Jews. But if you go to other places in the world, and even in the United States, you won't. You know, so, and you go to South Asia, you're going to get different subgroups, you know, generating. So yes, it's always going to be a function of who's included in the sample. So I think what you have to pay attention to is what the diversity of your particular sample is that you're trying to analyze and do this work in, and make sure that it's got representation, you know, in terms of the reference that you're using in some way.
Yeah, I agree. And that, but I also struggle with, like, you know, at what's, like, do we really care about the fine, like, you know, the PCs start getting into fine scale structure. Do we only care about continentals? Because at some point, like, you know, you don't really care whether
I mean, I was gonna say that, but I chose not to. You know, yes, there's structure within Europeans. You know,
Yes.
I mean, in the Middle East, right? I mean, Italians are not the same as Scots. You know, so.
Exactly.
Right? But do we care about that? And there's diversity of African ancestry. There's some diversity of Native American ancestry. So, yeah. So I think the highest, you have to be worried first about the highest level of diversity, of genetic diversity and so on. But that doesn't mean you should ignore, you know, automatically ignore what's within continents.
Yeah, exactly. Yeah.
Well, that was, Tim, that was a point of PC Relate, I think also. It's like,
Exactly.
you know,
Yeah.
to do final level structure, because if you don't, you could end up with problems.
Exactly. Yeah. Yeah. But like, but the problem is that, you know, it's difficult to interpret what the PCs mean, which is why we kind of stayed away from that, and we try to include proportional ancestry instead, because at least we know what that is. And so like you said, the PCs differ depending on who's in and who's not. And so we said, okay, let's avoid the PCs, kind of what, and then kind of do what kind of John was saying, is that let's use, instead of PCs, use proportional ancestry, because that should be universal and it doesn't depend on.
Usually they're highly correlated. I mean, in our experience, they're highly correlated, but not, you know, when you get to finer level structure. Okay, so the thing that's always amused me is like, okay, when we do Europeans, we use 10 PCs in the GWAS, but if we use other ethnic groups, it's 6. I don't know where this rule came from. But as you said, in Europeans, I think after PC2, they sort of become uninterpretable to me, at least in terms of geography. So I don't know what they're picking up. And in our experience, sometimes it's genotyping error. So I don't know, right? It's a little bit mysterious, you know, when you go to these higher level PCs, what they're actually denoting in your data, what structure. And I don't know if it's marriage structure, straight, you know, it's kind of, you know, diversity or what. It's very hard to interpret.
Yeah.
With the admixture, it's much more fixed. You know, with admixture, you're forcing an interpretation because you're saying it's due to continental, you know,
Exactly. Okay.
Okay, well, I would love to keep you longer, but I feel like we've overstayed our welcome as a group.
Appreciate it. No, this is fantastic. Thanks everyone for coming and hearing my talk. I appreciate all the feedback, and I learned a lot too, just thinking about things we need to focus on.
Yeah, well, we invite you back as a guest, so you can harass other speakers.
Appreciate
if you
that.
want, whenever.
I'll return the favor.
And we look forward to seeing more of your work.
Right.
Take care and
Thanks,
happy
everyone.
holidays.
Everyone.
Thank you, Tim.
Thank you.
Awesome. Thank you.
Thanks, everyone.